What's up guys, good evening my chilla party. This is your NSEJS Biology Master Teacher Akshansh and welcome back to the next session for NSEJS 2025 Biology Basic to Advanced series. We have already wrapped up one chapter, that is biomolecule, very important for your upcoming NSEJS 2025 exam. Now today I'll be starting with the chapter Cell, the Unit of Life. So cell, it is a fundamental building block of living organisms, and if we want to master biology, we'll have to start with the cell. So without any further delay, let's continue with the class. So since childhood, we have been studying that cell, it is a building block of all living organisms, right? It is a building block of life. Cell is the basic unit of life. Or we can say that cell is the structural and functional unit of life. Okay, so what is the cell? If I have to define cell, what is the cell? So cell is the basic unit of life. It is the basic unit of life. It is the fundamental unit of life. Why I'm saying it is the basic unit or it is a fundamental unit of life? Because no living organisms can have life without having a cellular organization. We must know that cellular organization, that means having a structure made up of cell, it is the defining characteristic of a living organism. So no living organisms can have a life if it is not made up of cells. Okay, so cell is the basic unit of life. No living organisms can have life without being cellular. Why? Because cell is the functional and structural unit of life. Okay, so cell, it is the basic unit of life. Why we are saying it has to be the basic unit or the fundamental unit? Because it is the functional and structural unit of life. Okay, so I'll write the next point, that cell is the structural and functional unit of life. It is the structural and functional unit of life. Do you know that all life forms, they start with a single cell? Being a unicellular organism, it is going to be made up of one cell only, and even for the multicellular organisms. So in junior classes, you must have studied about the process of fertilization. During fertilization, the ovum, it fuses or it gets fertilized by a sperm cell, now giving rise to the zygote. Zygote is a single cell entity. The zygote will further divide, it will give rise to the multicellular fetus. So eventually, the multicellular organisms, their life also starts with a single cell. So we can mention this point that all life begins as a single cell. All life begins as a single cell. Now, very important point. Now we can move and we can talk about the level of organization of the living organisms. So before getting into the depth of this chapter, I would like to tell you how the living organisms are organized, right? What is the structure? What is the level of the structural organization? So we are talking about the level of structural organization. Level of structural organization. Everything around us is matter. Matter is made up of atoms. This is what chemistry teaches us. So we human or all living organisms, we are also matter, right? So we must be made up of atoms. So with respect to the living organisms, the primary atoms that we have, basically they include carbon, hydrogen, oxygen and nitrogen, right? So the primary atoms that we are made up of. Okay, so the level of organization will start with atoms, and the primary atoms are going to be carbon, hydrogen, oxygen and nitrogen. These atoms, they come together to make molecules, right? So the atoms, they will give rise to molecules. So from atoms, we will get molecules. So what molecules? Let's say water, right? So we know that a major portion of us is made up of water only. So water, right? We will have sugars, proteins, etc. So basically we're talking about the biomolecules, right? We are talking about the biomolecules. So now after the molecules, we will get the cell organelles. Okay, so I'll write that from the molecules, we will get the origin of cell organelles. When I say cell organelles, it includes the nucleus, endoplasmic reticulum, Golgi body, ribosomes, etc. Right? So from the organelles will come together and they will make up a cell. Now, anything less than a cell, is not going to have any independent existence, nor it will be able to perform all the essential life processes. So NCERT says that anything less than a complete cell is not going to have an independent existence, nor it will be able to perform any of the essential life processes. This statement you have to remember by heart. So if you look around yourself, the very basic living organism that we have are the microbes, right? They are made up of single cell only, so they are unicellular entities. So anything less than a cell is not going to have an independent existence, nor it will be able to perform all the essential life processes, right? So now from this cell, now we'll talk about the multicellular organisms. Okay, from the cell you will get the tissues, right? So related cells, they will come together to make the tissue, right? So the tissue, they will make up the organs, right? So from the tissue you will get the organs. The organs, so related organs, they will come together to make the organ system. Organ system. And the organ system will actually give rise to your organism, right? So this is the level of structural organization. So in this chapter we are going to be primarily focusing about the cell, very important. Now we have completed the level of structural organization, so we can talk about the classification of living organisms on the basis of the complexity of their structural organization. So on the basis of the complexity of the structural organization, the living organisms can be classified as unicellular organisms or multicellular organisms. Okay, so let's move on and we'll write this thing, that on the basis of complexity of structural organization. The living organisms, they've been classified as unicellular or multicellular. So we can have living organisms they can either be unicellular or multicellular. So the unicellular organisms, they are capable of having an independent existence. The single cell is going to perform all the essential life processes. The multicellular organisms, they are going to be made up of multiple cells, right? So unicellular organisms, we can write some examples for unicellular organisms. We have the example of bacteria. We have amoeba. We have chlamydomonas. We have paramecium. Etc. Correct? Unicellular organisms. Let's talk about the complexity of the multicellular organisms now. So multicellular organisms, fungi except yeast, you will have the plants, the animals. They are going to be the multicellular organisms, right? So let's talk about the example of humans. So we humans, we are actually talking about an infant baby. So an infant baby is going to be made up of 2 trillion cells, right? I can write down some figures, right? So infant human baby, right? Not I should say infant only. So in human, the infant is going to be made up of 2 raised into 10 to the power 12 cells, right? That is 2 trillion cells. Now an adult, which is somewhere around the body weight of 60 kg, it is going to be made up of 100 trillion cells. 100 trillion cells, right? That is 100 into 10 to the power 12 cells. Itself in the human body, we have around 200 types of cells. So in human body, we have somewhere around 200 types of cell. So we can take the examples such as RBC, WBCs. Now WBCs have their own types. The cells making up the connective tissue, muscular tissue, nervous tissue, the neurons, nephrons, etc., etc., bone cells, cartilage cells, etc., etc., right? So we have more than, like, I should say, we have somewhere around 200 types of cells, different types of cells that are there in the human body, right? And an adult having weight somewhere around 60 kg, it is going to have 100 trillion or 100 into 10 power 12 cells, correct? I hope it is clear till here. Everything is clear till here. So now the next topic, that is about the discovery of the cell, how the cell was discovered. Okay, so we'll move on to the next page. Yes, so the topic is the discovery of the cell. So this cell, it was actually discovered by Robert Hooke. Robert Hooke. The cell, it was first discovered by Robert Hooke. So the cell was discovered by Robert Hooke in the year 1665. Now he was a mathematician and physicist. He was trying to develop his own primitive microscope. Well, he placed a very thin slice of cork of Spanish oak under his own microscope, and he observed some honeycomb-like structures, which he named them as cellula, right? Cellula or cell, that basically refers to the compartments that were there. So initially he wasn't aware of the discovery that he had made. He mistook it as like some structures meant for conducting fluids, but it was actually cell, and they were the dead cells. Okay, so I'll write this thing, that Robert Hooke in 1665, he placed a very thin slice of cork of Spanish oak. Cork of Spanish oak under his own primitive microscope, and he observed some honeycomb-like structure. Honeycomb-like structure, which he named as cellula. Latin word, it's a Latin word, cellula or cell. Correct? So very important, and you have to remember this thing. So he published all his findings in the book Micrographia. So if you want to remember the name of the book as well, so the book named was Micrographia. Please remember this. Okay, so what he had observed? He had observed dead cells actually. So this term cell, it actually means hollow compartment, right? What it means? Hollow compartments. And if you look at the cell, dead cells are hollow compartment because they have the dead protoplasm, right? But living cell, they are never hollow, right? They are going to have the plasma membrane. If it's a plant cell, it is going to have the plasma membrane, and out of it you will have a cell wall, and inside of the cell it is going to be filled with cytoplasm. So the term itself is misleading. The term cell is misleading, but we call them as cell only, right? So technically the term is misleading. It's a misnomer. It's a misnomer. So next, we'll talk about the discovery of living cells. So the living cells, they were first observed and sketched by Antony von Leeuwenhoek. By whom? Antony von Leeuwenhoek. So I'll just write Leeuwenhoek. It was Leeuwenhoek. Leeuwenhoek, who observed living cells. Who observed living cells. And not only he observed living cells, he actually drew the diagrams, right? So the diagram for RBC, for sperm cell, for protozoan, etc., etc. Next, Robert Brown. So Robert Brown in year 1831. Robert Brown. So in year 1831, he discovered nucleus. He discovered nucleus in the root cells of orchids. Nucleus. What was the sample taken? So cells of orchid root. Correct? So this is what you have to remember, right? Next topic, that is the cell theory. So the cell theory was formulated by Skleden and Shawn. The cell theory was formulated by Skleden and Shawn. So we'll talk about Skleden and Shawn first. So let's talk about Skleden. So Skleden, he was German botanist. Skleden, he was a German botanist. So Skleden, he was a German botanist, and in the year 1838, like during that time period, he was working with different type of plants, right? He was doing his research on different types of plants, and he came up with a conclusion, he came up with a hypothesis that different plants, right, they are made up of different types of cells, and the different type of cells are organized into different types of tissue, right? So I can write that Skleden, being a German botanist in year 1838, he said 1838, he observed and said, he observed and said, all plants are made up of, or I can simply write composed of, different types of cell. Different types of cells which further form tissue. Correct? Very important about Skleden. Now, after Skleden, Shawn. So Theodore Shawn, he was a British zoologist. Theodore Shawn, we are talking about like year 1839. So he was a British zoologist. So being a zoologist, Theodore Shawn, he was a British zoologist. So being a zoologist, he was working on some animal samples. So he was the first who reported the presence of plasma membrane outside the cell. So during his work, he also observed some plant tissue samples, right? Some plant samples. He observed that in addition to having the plasma membrane, the plant cells, they also have cell wall. So please remember that the cell wall was actually reported by Shawn, Theodore Shawn. Okay, so I'll write this point that Shawn, he observed animal cells and reported the presence of plasma membrane or cell membrane. And he also observed that the cell wall is peculiar to plant cells only. So that's how we differentiated, right? So if a cell is having a cell wall, it is going to be a plant cell, and if it is not having a cell wall, if the cell wall is absent, it is going to be an animal cell. So he was the one who differentiated between the plant cell and animal cell. Now we can move on to the next point. So now Theodore Shawn and Matthew Skleden, what they did, they combined their findings and they came up with a hypothesis. They said that all living organisms, they are made up of cell and their byproducts. Okay, so Theodore Shawn and Matthew Skleden, they came up with the finding that all living organisms are made up of cells and their byproducts. Okay, so this was the hypothesis that they proposed, and later on they proved it, but they were not able to justify from where these cells originate. So in year 1855, Rudolf Virchow, he came up with this finding that omnicellula acellular, that the cells originate from pre-existing cells, right? So the statement from the Rudolf Virchow actually wrapped up the whole cell theory, and the cell theory can be, like the final cell theory can be formulated as that all the living organisms, they are made up of cell and their byproducts, and these cells originate from pre-existing cells. So this is the final cell theory. So I'll write all these points for you. First of all, that Skleden and Schwann, they came up with a hypothesis that the living organisms are made up of cells and products of cells. Okay, and in year 1855, Rudolf Virchow came and he added omnis cellula e cellula. That is, cells originate from pre-existing cells. Okay, so we can write the final statement of the cell theory, right? I'll move to the next page. So the final statement of the cell theory can be stated as: first, all living organisms are made up of cells and their byproducts. Next, that cells originate from pre-existing cells. Okay, so here you have the cell theory. This is a very important topic and you can expect a question in NSEJS from this topic. Okay, so now let's continue to the next topic. So the next topic is, we are going to talk about the factors that limit the cell size or the cell shape, right? We will be talking about the factors that limit cell size or shape. So now it's a very important and very conceptual topic, because I'll be teaching you about the parameters, the factors that are going to influence the cell size and hence their activity, right? So the very first factor that limits the cell size is the nucleocytoplasmic ratio. The very first is nucleocytoplasmic ratio. So now let's talk about the situation. Let's say I am the mayor of a very small city. So since the place is very small, it will be easier for me to have my administration over it, right? To have control of everything, right? I can easily monitor all the activities that are going on in my city, right? If it's a small place. But let's say it's a very huge city, like Delhi or Mumbai, right? And if I'm the single person monitoring everything, it is going to be very hard for me. So the nucleocytoplasmic ratio, if the nucleocytoplasmic ratio, that is the volume of the nucleus divided by volume of the cytoplasm. So if you're going to have a very high nucleocytoplasmic ratio, that is higher the volume of nucleus and lower the volume of cytoplasm, the nucleus is going to have a better control over the cellular activity. So the nucleocytoplasmic ratio determines or it limits the cell size. Very important. Okay, so I hope this parameter is clear to all of you. The first is the nucleocytoplasmic ratio. The next is ability of oxygen and other materials to reach the various parts of the cell. Okay, so ability of oxygen and other materials to reach every part of the cell. Right, so oxygen required for respiration, other substances, other materials. They can include anything which are essential for the activity of the cell, right? So these parameters, right, the reach of oxygen or these material is also going to limit the cell size. Next is the ability of the waste substances, of the waste products to pass to the outside, right? How easy it is to remove the waste product from the cell. So now this parameter is also going to influence the size of the cell. Next is the metabolic rate, right? The rate of metabolic activities. So rate of metabolic activity, and the last parameter is surface to volume ratio. The next parameter is surface to volume ratio. So these 5 parameters are actually going to influence, or they are going to limit the cell size or the cell volume or the cell shape. Next, very important point from where you can expect a question in NSEJS, that the smaller cells or the metabolically active cells, they have very high nucleocytoplasmic ratio and very high surface area to volume ratio. Okay, so please remember that if a cell is active, it is always going to be smaller in size. Okay, so the active cells, they are going to be smaller in size. Why? Because being smaller, it allows the cell to have a very high nucleocytoplasmic ratio and a very high surface to volume ratio. Okay, whereas the cells that are passive, right, the passive cells such as ovum, which has very limited metabolic activity, right, let's say the passive cells, so they are going to be larger in size. Okay, if a cell is larger, it is going to be passive. So is it possible for a cell to be large in size and metabolically active? Yes. So please remember, if you have a cell which is large in size and it is showing good activity, right, it is very active, so it is either going to be cylindrical in shape or it is going to have some extensions such as microvilli. Okay, so if we have a larger cell with high activity, so how a large cell can achieve this? So it is either going to be cylindrical in shape or it is going to have microvilli. Very important. Okay, so you can expect a question from this topic as well, right? The factors that limit the cell size or cell shape. Let's move on to the next topic, that is about the various type of shapes of the cells that we have and the sizes. So we'll be talking about cell size and shape. I'll be talking about the examples that are given in NCERT, very important. Okay, so I'll write the cell and the respective feature. Cell, and here we have the features, right? So the first example that is given in NCERT is RBC. So RBC, let's change the color of the pen. Okay, so cells, features. The first example, let's take the green color, is the RBCs. Okay, so RBCs, they have biconcave shape, and their diameter is somewhere around 7 micrometer. Diameter 7 micrometer diameter. Okay, then WBCs, so they are amoeboid in shape. That is their feature. They are amoeboid. Okay, then we have the columnar cells. So they are long and narrow. Columnar cells, they are long and narrow. Then we have nerve cells, right? So if you check NCERT, the diagramming of nerve cells are given. Okay, so they are branched and long. They are branched and long. This is what the NCERT writes. Okay, they are branched and long. Then we have the mesophyll cells in the plants. Okay, so mesophyll cells. So they are round and oval, round to oval. So now typical eukaryotic cell, the NCERT mentions the typical size of a typical eukaryotic cell, right? So eukaryotic, we're talking about the typical eukaryotic cell, right? And generalized one. Okay, so the size is going to be 10 to 20 micrometer. 10 to 20 micrometer. We are talking about the size. Okay, then of a typical prokaryotic cell, typical prokaryotic cell. So the size is going to be somewhere around 1 to 2 micrometer. 1 to 2 micrometer. NCERT also. So this is actually about the size, right? The size. Talking about the length, bacterial length. So bacteria, they generally tend to have their length between 3 to 5 micrometer. Okay, this is what the NCERT writes. Okay, so I don't have space here. I can write it here actually. Is it visible? Yes. So in case of bacteria, we are talking about length. Okay, so it is 3 to 5 micrometer. This is length. So size and length, the data differs little. So size is going to be, obviously the bacteria, they generally tend to be longer in length, shorter in their breadth. Okay, so the size is going to be in between, right? So that's why you have less size. The size is going to be less, but the length, it varies, right, between 3 to 5 micrometer. Okay, so I will give you a quick revision of what we have learned till now. So first we talked about cell. Cell, it is the basic unit of life. It is the structural and functional unit of life. Then we talked about the level of structural organization. Okay, so we have the atoms. They come together to make the molecules. From the molecules, you get cell organelle, cell, tissue, organ system, organisms. Anything less than a cell is not going to have any independent existence, or nor it will be able to perform all the essential life processes. Now, on the basis of complexity of structural organization, living organisms, they can be classified as unicellular organisms or multicellular organisms. So bacteria, amoeba, chlamydomonas, paramecium, etc. These are unicellular organisms. Multicellular organisms, we can take the figures for human, right? So infant human, 2 trillion cells. The number rises to 100 trillion in the case of adult, weight about 60 kg, right? And in human body, you have more than 200 types of cells. Next, we talked about the cell theory. We talked about the discovery of cells. So the cell was discovered by Robert Hooke in year 1665. He observed a thin slice of cork of Spanish oak under his own self-made microscope, where he observed some honeycomb-like structures. He named those structures as cellular or cell, right? Then Leeuwenhoek, he observed the living cells. Robert Brown in year 1831, he observed nucleus in the cells of the orchid root. Then cell theory. So Schleiden and Schwann. Schleiden, he was a German botanist. He observed and said all plants are composed of different types of cells which further form the tissue. And Schwann, so he was a British zoologist. He observed animal cells and reported the presence of plasma membrane or cell membrane. And he also observed that cell wall is peculiar to plant cells only. Then they both came up with the hypothesis that the living organisms are made up of cells and products of cells. And later on they proved it as well, which came as to be the cell theory, but they were not able to explain how the cells originate, right? What is the origin? So then in year 1855, Rudolf Virchow, he came and he completed the cell theory. He said omnis cellula acellular, that the cells, they arise from pre-existing cells. So we can come up with the final statement of cell theory that all living organisms are made up of cells and their byproducts. Cells arise from pre-existing cells. This is the whole of the cell theory. Now then we talked about the factors that affect the cell size or shape. So nucleocytoplasmic ratio, ability of oxygen and other materials to reach every part of the cell, ability of waste products to pass to the outside, rate of the metabolic activity, and the surface to volume ratio, right? So active cells, they are generally smaller. They have a very high nucleocytoplasmic ratio and surface to volume ratio. Passive cells, they are generally larger in size. Larger cells with high activity. So how a cell can have a large size and as well as a high metabolic activity? So it is either going to be cylindrical in shape and or it is going to have extensions like microvilli. Okay, then cell size and shape. So cell RBC, so they are biconcave, 7 micrometer diameter. WBCs, they are amoeboid in shape. Columnar cells, narrow, long and narrow. Nerve cells, branched and long. Mesophiles sound round or to oval. Eukaryotic, the typical size ranges between 10 to 20 micrometer. Typical prokaryotic cell 1 to 2 micrometer, and the bacterial length, it is 3 to 5 micrometer. This is what the NCERT mentions. Okay, so that's all from my side for today's class, guys. We'll meet in the next class where we'll continue with this chapter and I'll teach you about the prokaryotic cell system. So we are done with the introduction to cell. Now we'll start with the prokaryotic cell, then we'll jump to the eukaryotic cell. We'll talk about all the organelles, and that's how we'll wrap up this chapter. Thank you very much. We'll see you in the next class. Good night, take care, and if you have any doubts, you can mention them in the comment section. Good night, take care. See you in the next class.